we are always beyond grateful that the Just Cancer Research Fund, led by the Stutmans and all the people who do the hard work to raise the funds and who dig in to support the research, come through even at times where that's not so easy, because as I'll try to summarize, it is indispensable for our research to find drugs that are more effective as treatments for GIST. And what I'd like to do is just describe quickly what some of the members of our group are doing and have done in the past year. We have a mix of folks in our lab, some of whom are here for a long time and some of whom come for a year or two from other countries, and they have an interest in GIST research. They want to learn techniques and make advances so that when they go back home, they can do better with their own laboratories to develop more effective therapies for GIST. Nacef is truly remarkable because he's the green thumb of the laboratory. The common theme in our laboratory is we need better drugs for GIST. Of course, with Gleevec and with the other drugs that our groups at Dana-Farber and Brigham have helped approve, we've done better in treating GIST, but we still need much better drugs. And without being able to grow the cells that come from the GIST surgeries and then treat them with drugs, it's simply not possible to find those. So Nacef is the one who has mastered the art of growing GIST cells from the patients, something that very few people in the world can do. And then Alexandra is the master at defining new mutations in GIST cells. I think you all know that part of the success in treating GIST is that the KIT mutation gives us a target for drug therapies, but we need more targets, and Alexandra is the best at defining new mutations that nobody knows about in GISTs that we can then attack with new drugs. Meijun is the pillar in our laboratory in understanding what we'll refer to as the biology of GIST. When we find mutations, those are in genes, and genes then make proteins, and the proteins interact with each other in the cells. They cause the GIST cells to grow, to survive. And if we understand the biology in the right ways, we can identify new therapies for GIST, and this is what Meijun does. We have Bin, who is a very well-established GIST specialist from China. She's with us for 2 years and is then going back in a few months to move ahead with the GIST research in her own laboratory. We need more targets in more than one way in GIST. By targets, I mean things that we can attack with drug therapies. And of course, beyond KIT, the central target, we need others. So one of the very important research pathways is to ask, how does KIT communicate through the cell? What are the so-called pathways? And Bin is the one in the laboratory who is breaking down those pathways. She's studying on a pathway that happens to be called NF1-ROS. And then we're finding new therapies based on that information that will better destroy the GIST cells. Stacy is part of the group who is, again, finding vulnerabilities in the GIST cells that are susceptible to therapy. One of the things that we're generally excited about in the group is we recognize that although KIT provides what's called oncogenic energy that makes the GIST cells grow, makes them survive when we don't want them to, it also creates some problems for the cells. It creates a kind of stress for the cells. The very thing that makes the cells move along and grow in ways that we don't want also creates some vulnerabilities, and Stacy is doing a lot of work to take those liabilities, a kind of double-edged sword, and use them against the GIST cells with new therapies. We have Armelle, who is another GIST specialist, this time from France, again has been with us for a few years and will be going back to her own research group in the near future. Armelle is doing some fascinating work where our lab has found that a gene that plays a critical role in muscular dystrophies, that's when you're born and you develop muscle weakness. There's lots of work that's gone on, sponsored by the government, for instance, to develop new therapies for those disorders. And we found that the same protein, it's called dystrophin, is mutated in GISTs to allow them to metastasis, the very thing that makes GISTs life-threatening. And Armelle is taking that information and moving that along to better therapies. Wenbin is a senior scientist who has a remarkable GIST lab in China, but he spends part of each year with us. And Tasos is an oncologist like Armelle who has his own GIST patients. He's from Greece, but again, he wanted to find better treatments for GIST, so he's working in our research laboratory. And what Wenbin and Tasos are doing, building upon Wenbin's work over the past 10 years really, is asking the question, we know that some GIST cells lose their dependence upon KIT, the central oncogene that we all know we attack successfully with Gleevec. And at that point, the cells don't respond clinically to Gleevec anymore. They become resistant. So Wenbin, with Tasos' help, has done a terrific job at telling us when the cells no longer care about KIT and they're no longer driven by KIT, what else are they driven by? What is the new motor? And how can we use that for better therapies? And they've moved this along to really a remarkable story that will be published soon that opens up new therapeutic opportunities. So I know that was a handful, but what I wanted to do was several things. One, give a sense of the commitment of the laboratory to finding new therapies for GIST, and also to let you know that all of the eggs aren't in one basket. We have several research programs going along, realizing that science is unpredictable, but life is precious, and that by attacking several of these at the same time, we can be sure, just as we've developed effective drugs in the past, that there'll be more in the next few years. So again, thank you so much, GIST Cancer Research Fund, all the supporters who make this possible, for allowing us to do research to find cures for GIST.